As I've already described for you, the thalamus provides the principal input that modulates activity in cortical networks. That activity goes into the middle layers of the cerebral cortex, layer 4, and from there it's amplified. So one purpose of this network of radial connections is to amplify this signal. So I've already mentioned that our thalamus sends inputs up to layer 4, providing the main source of drive to this network of cells that populate the cerebral cortex. There's also a small input to layer 6, but the primary one that seems to have the greatest physiological impact is destined for layer 4. And as you draw, you can hold yourself accountable to what you actually see from your own hand. So what you produce is what you can then interrogate. And here's how you might go about interrogating your drawings. Let's imagine that you select the left medial lemniscus. Okay, so I'm placing my cursor here on the left medial lemniscus. You might ask yourself questions like this. Where are the cell bodies that grew these axons? Or you might ask, where are the synapses of these axons? Now, I'm going to on the medial aspect of the internal capsule and bulging in to the lateral wall of the lateral ventricle, we have the caudate nucleus. And then where the caudate nucleus and the putamen join together, inferior to the anterior limb of the internal capsule, that's what we call the ventral striatum, or in this region, primarily we call this the nucleus accumbens. Now, I'd like to emphasize that the basal ganglia is all about modulating movement. And what happens here is that there are two anterior cerebral arteries, one that gives supply to the left hemisphere, one that gives rise to the right hemisphere. And if I gently reflect away the optic chiasm, I think we can see those arteries as they come together. And there is a short anterior communicating artery that connects the two anterior cerebrals. So just in front of one interesting point about the central sulcus is that sometimes it can be very difficult to find in a human brain because the brains often don't look like the simplified figures we often see in textbooks. But there is a way to find the central sulcus.